I didn't do enough of the actually.
All right. Good morning, everyone. Everyone who's here, and to those who may be following this that we're live streaming. Thank you for joining us. Some of you may have been here. Some of you were here a little over a year ago when we did our first meeting on this topic of neurotechnology here at UCLA. And the takeaway from that meeting was that we should meet again, and we should meet on a semi-regular basis, which we took to interpret as an annual basis on a forum like this, although we also have been having monthly or bi-monthly meetings with different speakers from within UCLA, so look out for those announcements. I think we all recognize that neurotechnology is at a point of significant growth. I won't use the word exponential because that's I don't know exactly what that means, but it's a growing field with a lot of new technologies being developed, a lot more interest both within academia and in industry, and it's really a broad and inclusive field. So I, as a neurosurgeon and a neuroscientist, have a strong interest in it because as a clinician I implant brain stimulators to try and improve function. But the work that I do as a clinician couldn't happen without the neuroscience behind it and without the engineering to develop the technology. Now, unfortunately, from a clinical perspective, the clinical engineering is a little slower to develop than we all might like, and translating what is really cutting-edge neurotechnology from the lab to the bedside or to the operating room is a little slower than we'd like. But the goal of this type of meeting is to bring all of us into a common forum where clinicians, neurosurgeons, neurologists, rehab physicians, as well as engineers, be it what if you call yourself a neuroengineer, an electrical engineer, or a materials engineer, and neuroscientists all get to talk together and interact and share with each other what we are doing. We purposefully designed the program into different segments with at least pairs of speakers or 2 to 3 speakers, so we hear different perspectives on similar topics. So for example, the session that I'm going to speak in is on visual prostheses, and I will share what I've been doing, but we also have Dr. Wentai Liu, who is a professor in bioengineering, who has done tremendous work in this area. So you'll hear two different perspectives and how our areas will intersect. And we're hoping we'll highlight these intersections and promote greater interaction. I don't know if you want to make a couple comments, Nadia. No, she doesn't.
No, I mean, you know, like you said, we're very excited to have you all here, because I really think that this is a great opportunity, an interdisciplinary opportunity. And we also have students, junior faculty, postdocs, very senior faculty. So I'm hoping that we can, you know, have some good interactions, good discussions about interdisciplinary work, and come up with some great future projects that we can, you know, foster in this kind of forum. So.
Yeah, and so I'll make one other statement, since we have a few minutes. One of our takeaways from the last meeting we had was to discuss how we continue this interaction, how we make it grow. Besides having meetings like this, besides having monthly seminars, and we all decided that students are an outstanding bridge to make that happen. And that perhaps one of the important cornerstones of this program should be to have an interdisciplinary training program. And I wanted to update everyone that Nanthia and I have been in close discussions with the NIH. Obviously, you don't just discuss things with the NIH to get awarded, but we've been working very diligently to learn about what it is that makes a successful program, learning from other programs across the country. I think University of Pennsylvania has a very successful program, and others do as well, to really optimize our opportunities to enhance this interdisciplinary training. I think it also, you know, I was just having a discussion before we started this meeting that all of us are really busy. Many of us have many grants that we're already working on, and to stop and try and build bridges is sometimes difficult, but students are often the best way to try and initiate that bridge building. So we're hopeful that that will do the same. I also want to acknowledge that we have been seeking industry support. It's been a little bit slower than we'd like, but I do want to acknowledge Medtronic that did give us a $25,000 grant to support our Center for Neurotechnology. We haven't spent it, so don't think that we've been doing anything nefarious. What's that? Today. We're using some of it to support today, like the live streaming as well as the food. But we want to, besides building these bridges, I'll use the word again, here amongst us, we want to build relationships with industry, because industry really, they have resources that we don't. They have perspectives that we don't, and they actually value our perspective and resources and innovation as well. And I think it's an important relationship to foster and build further. So the one final acknowledgement is this is supported by a grant, or the Brain Research Institute provides funding through affinity groups that apply for, so we applied as an affinity group. Actually, Nanthia did the application for an affinity group to foster interest in neurotechnology, and this is supported by that as well. All right, so any questions before we get going? All right, otherwise we'll get started with the first series of talks, which is, so they're broken up into 40 to 1 hour minute sessions. First one is on spinal modulation for rehabilitation, and we have two speakers, Dr. Daniel Liu and Dr. Reggie Edgerton. Dan is associate professor in neurosurgery and has been doing work both with invasive and non-invasive modulation, primarily focusing on upper extremity function. And then Reggie Edgerton, who you all probably know, has been working in this field for a long time with animal models, and if you saw some recent press about people who are being rehabilitated, I think all of that is really premised on the work that Reggie did to get it into a clinical trial. So, you know, another nod to all the outstanding work that's going on at UCLA. So we're looking forward to the talks. I think Dan is going to speak first. Thank you very much.
Thanks, Nader, for that introduction. Obviously, my time with Reggie, who is a close collaborator with this, the first part of the talk is our work on upper extremity function, and later on we'll talk about the autonomic function as well. So as a neurosurgeon, you know, I publish a lot on how to do surgeries to treat spinal cord injury and conditions like that. We instrument, we put in a lot of effort. Our 7 years of residency training is directed towards how, you know, exactly how to do that. And there's a lot of effort and a lot of money spent in just the stabilization aspect of it when the patient just comes in to be stabilized. But in terms of all that effort that's actually put into that, it's actually pretty disconcerting in terms of how it does not really impact on the recovery of the function of these patients. So if you look at this, which was published in 2008, these are recovery curves based on their injury grade. So it goes from ASIA A to all the way to ASIA D. In patients who are ASIA A or B, who are motor complete, you can see that their injury recovery curve is actually pretty dismal. There's really not much of recovery of function after initial injury and stabilization. So we're spending a lot of money up front here, but really not a whole lot out here. And really currently the treatment paradigm is that the decompression and stabilization in terms of what I do as a neurosurgeon really does not improve function. And this is, you know, borne out in this Bible that we actually all use in neurosurgery, which is operating on a patient with complete spinal cord injury does not result in significant recovery of neurological function. So we essentially, you know, the message here, at least for us, is that, you know, you can operate on them early or late or whatever manner you want. There's really no recovery of function. It doesn't impact on the recovery. So the main problem, I think, to look at this is that the spinal cord is actually very, very high real estate territory. Within this organ, which is about ½ an inch, there's probably 50 anatomical tracts, 1 billion neurons, and 7 trillion connections. There's countless descending and ascending tracts, efferent and afferent tracts. And injury of these tracts would cause substantial dysfunction. And this, just as kind of comment on stem cell therapy, you know, currently the stem cell therapy as it stands now, it's really difficult for me to imagine that it's going to reconstitute all these spine tracts all in this, you know, ½ an inch territory. So looking at this from the other side of the coin, which is that it is very elegant and eloquent organ. However, the spinal cord injury is never anatomical complete. And novel treatment may reinforce alternative pathways because there are so many tracts and efferent and afferent pathways that there may be potentially preserved pathways, and that may potentially serve as ways to re-enable spinal cord function. And then the other observation that spinal cord is smart and automatic, and complex motor behaviors of extremities are largely controlled by network within the spinal cord with minimal input from the brain. And actually, Reggie likely will speak to more of this aspect. And he's one of the pioneers in that spinal cord mechanism and circuit related to motor function. So looking at this from this perspective, you look at it, after spinal cord injury, you do have substantial loss, but there's actually substantial spare tissue as well. In the spinal cord injury setting, it's never anatomical transection. You never often see that. You do see that, you know, in bullet wounds or knife wounds, but oftentimes it's not a complete transection, and you do have spared areas. And these are spared tracts that can potentially lead to attenuated signal from the supraspinal aspect that can be either modulated to be increased, or the receptors or the receptive motor neurons can actually be further activated to actually execute a movement. The first topic that I'd like to address is the hand function. And why study hand function? Because the majority of patients suffer from tetraplegia. And restoration of hand function is cited as one of the highest priority in spinal cord injury surveys. Improvement in arm and hand function observed within the 6 months time period. However, there's minimal improvement after 1 year after physical therapy and training. And the hypothesis that I'd like to address is whether cervical motor network related to upper extremity function can be accessed by epidural stimulation in spinal cord injury to improve motor function after spinal cord injury in patients. So epidural stimulation is an FDA-approved device that is implanted in patients with chronic pain. We've readapted this device for the treatment of patients who have a spinal cord injury. And this involves implanting electrodes in the cervical spine and also a battery that's actually remote controlled into the flank or the abdomen. So we selected 2 subjects that have spinal cord injury that's non-transected and have, on the axial scan, it shows that there's sparing of tissue. They're both motor complete but sensory incomplete patients. One is C5, another is C6 level of injury. And we use this hand grip device to test their hand function. And these are various EMG devices that we can capture their electrical activity of their muscles during a contraction. So these 2 subjects are implanted. You can see the electrode posteriorly there and there. These are the instrumentation that they had for their treatment of their spinal cord injury for stabilization. These highlighted gray areas are the anode and cathode pairs that we actually mapped out to show that they're able to access the circuit for upper extremity function. And here you can see that these various different muscles, that hypothenar, flexor digitorum, biceps, in various different configurations. These are the pairs that we use, minus 11 and 12, shows that 15 and 6, actually 13 and 14 shows most robust recruitment of these muscles in the hands. And so that's a pair, electrode pair that we actually use. So these are dose response of the location, and we increase the intensity, showing that there's actually evoked responses from these motor pools, showing that potentially, if we target these areas, then potentially we can improve function in the hands. And the same goes for subject 2. We did the same mapping strategy to obtain that. So using this electrode pair, we actually applied it in a chronic setting to the patients while we do rehab training on the hands. And so we mapped out what the optimal frequency is and intensity for this particular subject. And you can see that the force, we have to ask the patient to generate the force in the hands, and you can see that they're at 20 or 30 hertz, they're able to generate probably the most optimal, maximal force. And then we also mapped the intensity that showed by 1 or 1.3 milliamps demonstrated good response. And that's the second subject. We also asked them to trace the sinusoidal wave to look at their accuracy, and again, 20 hertz appears to be the most optimal. And then in terms of response time, these patients actually have delayed response to a command to activate their muscles, and it demonstrates that in terms of frequency, 20 to 30 hertz actually showed substantial decrease in the response time. So given that, we actually trained them. So we used 30 hertz frequency at 1.0 milliamps and trained them throughout this period of time. This is sham training, in which we imparted about 5% of the stimulation and demonstrated that they're actually pretty stable in terms of their hand function. And during this period of time, they actually received a lot of rehabilitation intensively. And after 14 weeks, actually, we subjected them to this paradigm where we actually stimulated them. And you can see that gradually you can observe that the force actually increased in terms of their ability, hand ability. And this is without stimulation at baseline. So their baseline ability, even without stimulation, increased substantially so. And then this is with stimulation, which is the curve, which shows that there's more improved gains. 20 newtons of force is usually the force that we would like to get the patient get past, because that allows them to grip an object, potentially turn the door handle, and whatnot. In terms of hand control, I alluded to this before, is that if you ask them to trace a sinusoidal wave without the stimulation, they're able to do it, but not in a very good manner. And while you actually ask them to, with the stimulation on, they're actually able to trace this wave much better. In terms of endurance, in terms of endurance, we actually asked them before stimulation, after stimulation, to to go above and below this line in as fast manner as possible, to oscillate above and below in a controlled manner, and they're able to do so with the simulation. And just a little bit about the mechanism. I won't go into too much about the mechanism. This is not a tonic effect where the simulation is on, you activate the whole musculature. This is actually a modulation effect to the motor circuit. And you can see that with stimulation, the EMG appears to be larger, and the pattern of stimulation is that it actually has its own morphology. So it's not an active tonic stimulation that activates a motor pool that causes a contraction of all the muscles. It's actually very discrete modulation that the motor pool is activated distinctively to the task that the patient's able to do. And this is the functional outcome. This is for those people who say, well, you're activating the hand function. Yeah, that looks great, but how does that impact on the daily living? And you can see that with the simulation on, at the conclusion of the study, there's substantial improvement in terms of the ASIA scoring system. And the spinal cord independence measure, which measures respiratory function mobility in toilet and outdoors, it shows a significant improvement. Now, tricep strength, which is C7, is a very important muscle in terms of these patients with spinal cord injury, because that allows them to transfer, use the wheelchair more efficiently. So you can see that getting 3 out of 5, or even in this case 5 out, 5 out from the strength of 2, which is below anti-gravity and twitches, to 5. That's actually pretty substantial. And that impacted, as he said, as we alluded here, the spinal cord independence measure scores of these 2 subjects. This is a picture. There's limitations of showing these pictures because it really doesn't tell you what's exactly being done on these patients, but nonetheless, with that in mind, this is him trying to grip, and it's a tenodiscus grip, trying to displace the hand grip, and it's very difficult. In this case, he actually has a little bit more intrinsic, able to activate the hand grip muscle. This is asking him to oscillate voluntarily, so he's activating the muscle, his muscles voluntarily above and below those 2 designated lines. So it shows that there's some improvement in terms of the hand function per se itself, and also in terms of the functional scores. And this is where I think the important part about this is that it not only makes for that good video, but it actually impacts on their motor score and also their function. In the spinal cord independence measure, you can see that this is the traditional curve of what a patient should be after spinal cord injury. And these 2 subjects, they're all about 20 weeks after injury. You can see that they actually are no longer in that type of curve, that recovery curve that we see. So, and both in the spinal cord, the ASIA score and the spinal cord independence measure. We moved beyond those 2 patients. We implanted 2 other patients under the auspice of the NIH grant. Currently, these injuries are all very different in every single individual. So we're finding out ways to properly map them and properly implant these and use the appropriate electrodes to improve the function. The previous study was not completely sham. In this case, it's a triple-blinded study in terms of seeing if there's an effect. And you can see that without stimulation, the force of this subject, where it was implanted, is below 5 newtons of force. During sham, it actually is about the same, and with treatment, under treatment conditions, with this mapped, using this AR4, CR5 electrode pair, there's improvement in the hand function. So it looks like we're designing algorithms to focus on what the residual motor pool looks like and residual connection looks like, and we're using that algorithm to see if we can get further improvements from these patients. So in summary, modulation of cervical spinal cord can be performed to enhance motor function of the upper limbs after chronic spinal cord injury. Neuromodulation of cervical spinal cord can enhance both strength and motor control. And the mechanism of action likely relates to increasing activation state of the spinal circuitry. And I'll leave Reggie to expound on that during his talk in terms of some of his studies and also the animal studies as well. So the sort of the downsides, I won't say downside, but currently it's not clear whether all this effort in terms of implantation and subjecting the patient to all this risk is worthwhile, simply because it is a surgery for these spinal cord injury patients. The patients are somewhat fragile. So we don't do this without that in the back of our mind, and we are always going after functional improvements, and that's certainly some of the focus for these type of studies and going forward. Is it worthwhile to implant and subject these patients to all this risk, all to just show that they can move their hands? And if they can move their hands, doesn't necessarily mean that that impacts on their quality of life at all. So that's something that we really need to keep in mind as we go through these, as you listen to talks throughout the day today. Just because we can do it and we can implant all these fancy brain-machine interface stuff, is that worth the effort? And so you really have to document that there's functional improvement and that the impacts on their quality of life, and that's going to diminish improve their independence and diminish their dependence on caregivers. So I'm going to switch gears to autonomic function. So because we can actually modulate something as complex as hand function, presumably in terms of autonomic function such as the bladder function could be something that's more easily modulated and potentially improved. Bladder function in spinal cord injury, there's essentially only two muscles that you have to focus on, which is the detrusor muscle, which is on top of the bladder, and the urethrosphincter muscle, which is actually the gateway to urination. So those are only two muscles that you need to be aware of in spinal cord injury. And after spinal cord injury, there's a condition called detrusor sphincter dysynergia, where both muscles are in spasm and they co-contract. When that happens, the urine goes to pathway of least resistance, which goes up to the refluxes up to the kidneys, and potentially could cause sepsis and kidney failure. So that's a pretty devastating condition. So the treatment currently is using catheterization. I'm just going to skip through a little bit of this, but you guys can certainly come up and talk with me afterwards. But bladder function is important. We published a study in which we are able to select about 5 patients with anatomically incomplete injuries, but motor complete injuries with this condition called DSD, or detrusor sphincter dysynergia. Essentially, these patients, through magnetic stimulation, non-invasively, you can actually induce micturition or voluntary urination through 14 weeks of treatment. They're actually able to urinate without stimulation on their own after that. So before stimulation, you can see that this is a urodynamic study showing the bladder filling, and you ask them to urinate, there's no flow through that. But after treatment, you can see that this is a urodynamic pressure diagram. It shows that there's actually now flow. I'll just skip to the last slide just to show you what happens with this. So these are 5 subjects in terms of the voluntary urination. This is an assessment period of time using low frequency simulation. You can see that after 4 trials, this subject actually able to demonstrate voluntary urination up to this point. And we did actually, the response is variable. There's a subject E that shows minimal improvement, some improvement, but not as robust as the other subjects. And you can see that the urine stream improves, the bladder capacity improves, the residual volume decreases, cell catheterization frequency decreases, and the sexual health inventory score for male actually improves. And the quality of life score also improves. So it shows that this non-invasive way of treating the spinal cord injury can be effective in this pilot trial. So these are ways of modulating the spinal cord without, I'll leave Reggie to discuss the potential mechanism, but this is a novel, non-invasive way without subjecting the patient to a lot of potential risks to re-enable function and restore independence and function in their daily lives. Okay. Thank you.
Where's
Go ahead.
Yeah. Hi. In terms of regarding the last study you did on bladder control, I was just wondering, I know it's 5 patients, so it's difficult to say, but do you have a hint on what could have been causing that difference in performance? Would it be the time since injury, or would it be like type of lesion they had? Again, I know it's 5 subjects, but I was wondering.
I think, no, it's too early to say, but the 2 best responders, one was 13 years from injury, and the other one was like 5 years. So it's not the chronicity of injury, unlikely to be that. It could be the amount of spare tissue, also potentially what their sensory preservation is. So there are several things that we're looking at right now. It's just too small of a sample group to derive any potential, and we are doing larger studies to address what exactly you asked. Who are the best patients that can respond to this strategy? Yes, Bruce.
Is this working? Oh, yes. So over the last 30, 40 years, a variety of ways to get people to take steps, standing and walking, who are paraplegic, people to make use of the upper extremity with cervical spinal cord injuries have come about. Going back to Wayne State, going back to the Power Walker, which basically stimulated 3 or 4 muscles in each leg, and allowed people to slowly take steps over ground. And the freehand, which put electrodes into the muscles that will allow for elbow flexion, extension, wrist flexion, extension, or intrinsic hand muscle function, depending on how many wire electrodes you put in. All these groups became commercial companies, and they all died. Because patients, it turns out, we're very excited, but as clinicians, about trying to increase actual function, not strength, but actual function. And a surprising number of people don't want to put up with the complexities of these things, the side effects, and in some cases, physicians are to blame for not trying to promote those things that you just don't know enough about. And these interventions are really complex and far more invasive, with well-known complications. So, for example, someone with a cervical injury, get epidural abscess, and you could lose 1 or 2 levels of function. It would be disastrous. So there are not only ethical issues here, but sort of trying to wrap your arms around what is it that most patients with these injuries will want. If there were 100,000 people who might have considered using the freehand, there were a couple of dozen who actually did it. And that wasn't nearly as invasive. I don't know what the answer to this is, but it's clearly something we have to be thinking about. Even achieving walking over ground, if it's very slow and effortful, you're still going to use your wheelchair most of the time. If regaining a little bit of extra strength in a hand that doesn't allow you to actually, bring the glass to your mouth or transfer better from your wheelchair isn't going to be very exciting to people, and will never be a way for us to promise that we'll be able to accomplish that. So I'm throwing this out, not to throw a wet blanket on things, but to remind us of the realities of people with spinal cord injury who are highly disabled. We only see the people who are really gung-ho, who are willing to move to a new city just to participate in a trial. There aren't a lot of those people, from what I can tell.
No, I fully understand that, and that's one of the reasons why we're going after functional improvement, and that's exactly it. Is that there's a lot of engineers in the audience right here. Just because you can implant brain-machine interface doesn't mean that you ought to do it. It doesn't mean that it may impact on the quality of life of these patients. So I fully acknowledge that that possibility, and certainly that's something that we strive towards to impact patients in a meaningful way without subjecting them to undue risks.
Thank you, Dan. So just to remain on time, we'll move on to the next speaker.
We gave Reggie a brief introduction before, but Reggie Edgerton is a professor in neurobiology. Which department are you in? He works across multiple departments. He's in neurosurgery. He's bioengineering, Yeah, I mean,
biology.
yeah, see, there's a lot that he
There's
does. But Reggie's really, he's been in this field, not to date him or age him, but he taught me when I was an undergrad here at UCLA, so he doesn't remember that, but so he's been at this a while. So thank you very much.
Well, the fact that I'm in so many different departments, that is telling a story in itself, is that maybe we shouldn't be in these departments, maybe we not have these boundaries. I'm going to approach this presentation from the standpoint of new scientists, clinicians coming into the area, and talking about a training grant. I think we really need a training grant in this area to help cross the borders. And so just think about that, what I'm saying relative to how we're going to prepare the new generation for what is to come. And I will say that in spite of the reservations, I think there's always been reservations, and there always will be. But when we say reality, we need to think, especially if you're just getting into the area, you need to think of what is reality today is not necessarily reality for the future. So that's another way of looking at it. Now, I would say we have, and colleagues, have found a number of, have made a number of observations which have been rather surprising. And the levels of recovery were ones that would not have been predicted based on the present dogma. And interestingly, I think one of the reasons that we've seen these things is that we've actually exposed a new physiology that's related to the control of movement and control within the autonomic system. And so this has occurred with old technology. We started this neuromodulation of the spinal cord with trying to get individuals to stand and to step. We did that with the old technology that's decades old that was used for pain. So we're in a state now that I think we need to get the technology updated to what we think is possible for the future. And one of the ways that we can help to make that happen is to have a successful training grant here and at other places. So what I'm going to do is talk just a little bit about how we think the neuromodulation is working, why it's working, and how it's taking advantage of a new physiology which we had not appreciated before. And then I'm going to go over several outcomes from this neuromodulation so you can get an idea of the potential impacts of neuromodulation if you use it in very specific ways. So I don't want to try to convince you that each of these observations is, you don't even have to believe them, but I'm just going to show you what the observations are, and then time will tell how effective and how acceptable they are. Another point that I would like to make in all these experiments when we started working with human subjects is we decided that the correct way to do this, or the most effective way to do these studies, is to study extensively and very quantitatively just a few subjects, rather than studying hundreds and getting relatively superficial information and giving you no idea of what is the biology behind it. So we are pretty convinced that the way to really find new things in this area, you need to focus on the details of a subject and get to know what the subject is really going through during those treatments. And then finally, I'm going to give you an example of why we think it's important for technology and biology to be working very closely together, basically as a single unit, sharing ideas. The example is very clear. So we're trying to develop technologies, and it's going very slowly. Now, when we basically got started in 2008, we had applied to NIH to implant the pain epidural stimulator from Medtronics to see if we could at least get to the proof of principle. Can we take advantage of this automaticity and smartness within the circuitry of the spinal cord? So this is what the general thinking was in 2008. And then I'll show you this slide and how I think it's changed. But I think the perception, I'm not saying that everybody is going to believe this, but from my perspective, this was the perception. But a lot of those concepts, I think we have to throw away and think of a new possibility. So, for example, the idea that Dan was talking about, the idea that recovery is not going to occur after 6 months. Well, all of the subjects that we've studied basically have been injured for more than a year, and some of them up to 20 years. So one of the key experiments was this one, and this does not even include modulation. But this shows an animal that was completely spinalized, could not step at all, had been spinalized for 3 months. We put it on the treadmill after we gave it strychnine, and the strychnine facilitated the circuitry by disinhibiting. It's a glycine receptor. And this animal, 30 minutes after we injected IP, this animal could walk full weight bearing over a range of speeds. So that tells us, and that's no input from the brain, and the spinal cord is walking like this. So this goes back to the basic concept from the beginning when we started doing the animal studies, is how much of the automaticity that's built into the spinal circuitry can we take advantage of after complete paralysis or severe paralysis. So this was telling us a lot. In fact, it was telling us more than we were even realizing. We said, well, this is great, but we keep understanding the significance of that. Now, combining with technology, after a number of years, and we had working with epidural stimulation, and the other is the transcutaneous non-invasive stimulation like Dan was talking about. And this is an example of a person that has complete paralysis. The fact that he's blind is just a side of interest, but he walks thousands of steps in this exoskeleton, and what we're hoping with this type of exoskeleton, it will tell you how much work the subject is doing and how much work the robot is doing. So it will do all of the work if we let it. So the question is, with neuromodulation and training, can we get the person to learn to step so that they become more independent of the robotics? And after 5 sessions, we'd already seen, this is with transcutaneous, non-invasive stimulation. We started stimulating him, and after 5 days, we tested him similar to what we had with the epidural implanted subjects who regained voluntary control, again, in chronic, complete paralysis. Each of the first four subjects that we studied regained voluntary control and movement similar to what you see here. Now, after 5 sessions, and this is, I think, approximately 3 years after the injury, had a very complex, relatively low thoracic injury, and we turned the stimulator, we asked him to move, and he can't move his legs. Turn the stimulator on, and he doesn't move. This is a key point. In fact, it's related to what Bruce was just talking about. Stimulator on does nothing. We are neuromodulating the circuitry. We're not inducing a movement. We're enabling the movement. And so it tells him to flex his knee, and he flexes his knee. This is the first time he had done it since he'd been completely paralyzed. And this is voluntary. So these are the type of findings that makes us think that there's things here to take advantage of. Again, what we're basically testing is what can happen. We're not testing what does happen in a large population. So we're finding out what can happen, and then we can take it from there as to whether it's going to be practical and so forth. Now, what's the mechanism? I'll give you just a short idea, but the first experiment we did with the first subject was epidural implant. This is exactly what we observed. And this observation shows that as we increased the voltage, got no force until we got to about 7 volts. And at 7 volts, the subject says, I feel connected. And I don't know anybody that knows what that meant, but he could generate a little bit of force, and as we increase the voltage, more force. And basically what we are doing, I think, is increasing the excitability of the networks that are generating this type of movement. And now Dan showed you this, and from the beginning we thought, well, this kind of fits this pattern. That is, you've got to reach this motor threshold, and then you can move as you wish. But after the injury, we don't know how much activity automaticity is remaining. And maybe you're completely paralyzed until you get to this point here. So with the enabling motor control, the electrical stimulation, if we put that, combine that with this, we can get above the motor threshold. Now, what's the nature of this connectivity? We don't know. I have no idea what the anatomical basis of it is, but we know for certain that there's physiological, there's functional connections that are formed very rapidly. And in fact, in some of the subjects with neuromodulation transcutaneously, some of the subjects showed some voluntary influence within 1 session. So that tells us one thing, that this lesion that we call complete paralysis, it needs to be re-examined, and we have no idea what the real biology is behind that. Now, to give you a little bit of a physiology here, this is, I want you to look at, I'll skip some steps here. So ordinarily we would say the resting excitability is, say, this level. This is the motor threshold. You've got to get to this motor threshold to get movement. And the more effort, you'll recruit more motor units and get more force. But we think after injury that resting level goes significantly down. There's a lot of loss of synaptic contact to these motor neurons down here. So, so we think that with the stimulation we can bring it back up, and then that enables the person to move. Now, this what happens. The major point that I want to make here. 2 minutes left. The major point that I want to make is that this physiology below the motor threshold is extremely important, and it's been ignored in electrophysiological experiments. This is an example of individuals with complete paralysis, and I'm going to move this on up. This is what we call 0G gravity or situation. We ask them if they can move voluntarily. They've had 18 sessions of treatment here. Again, complete paralysis, and then we turn the stimulator on. We ask them to voluntarily move, and then you add the stimulation to it, and this is the type of movement that you can see. So the bottom line is there's a lot of potential in the spinal cord that we have not known about, and this shows the force, the knee angles that have changed with just voluntary effort, and this voluntary plus stimulation. But a key point here is some of these individuals were able to move their limbs as well without stimulation as they were with stimulation. The point there is there's a plasticity that's occurring so that they become more independent of the stimulation. Will they ever become fully independent? That remains to be determined, and I would guess probably not. And so with hand movement, you've already seen that, and the type of improvement that can occur there. This is with eight treatment sessions, non-invasive stimulation over 4 weeks, 2 times a week, and this is their improvement in grip force. So with and without stimulation. So the difference between before without stimulation and what they could do with stimulation at the end was over 300% increase. And we have measures of that's related to quality of life and what they could do with their hands and so forth that is consistent with this is really functional movement. One individual has gone from not being able to feed themselves to feeding themselves, and I would say that has some impact on quality of life. Now, this is the example that I wanted to show you that show you why physiology and technology needs to be working together. This is the trained mice to step with a fixed trajectory versus not a fixed trajectory, but one that allows variability. This is untrained rats, spinalized, and you see the variability. If you train them, you see this, but even when they're trained, you see a lot of variability. The point is, the basic biology of movement has the intrinsic properties of this control is that there's got to be variation. There is significant variation. And so when we train them with fixed versus no variation, they step much better. So this is an example. The basic concept here is that if the spinal cord has decided the foot is going to be here versus here, the robot has already decided it's got to be here. So it's in a constant mode of correction. And therefore, what happens is that the reciprocity of flexors and extensors is totally disrupted. And so the spinal cord is being corrected constantly, so we can't learn what it's supposed to be doing. So there's one of the most successful robots on the market for stepping has a fixed trajectory. And that device serves as a coat hanger in many laboratories today because it is not taking advantage of a basic property of physiology. So, using neuromodulation, we enable, we don't induce. Those experiments that Bruce was talking about, they're implanted electrodes in the muscle. The muscle is not that smart. The smartness is in the spinal cord. So we enable the spinal cord. We take advantage of the control. So if you're directly stimulating the muscle, the movement will be only that of which muscles you're stimulating. But if you're neuromodulated and you've got the brain involved, then you've got your full range of movement that you can perform. You're not limited. So this is going back to assuming dominance of the cortical control. I think we know that that's not so great. It was not as great as what has been thought. Is it important? Yes, it's important. Can we do a lot without it? Yes. The magnitude of the automaticity, we're still impressed with how much that's the point. Proprioception, you saw that cat stepping. That was control only coming from proprioception and cutaneous information. And not realizing the role of activity-dependent plasticity, this is critical to the whole thing. You can activate the circuitry, but if you don't tell the circuitry what to do, it doesn't know how to adapt. It has to be given useful information, functional information to tell it how to adapt. So that's why it's so important. And the dogma that nothing can happen after a year, that's based on the results that we've seen, that's ridiculous. And we have to admit, we don't know what that lesion is for complete paralysis. We don't have a clue. What we have been assuming it is not correct. So I would say there's tremendous potential for the future. This is just the beginning. So if I was a new, young postdoc or that was interested in this area, I would say there's tremendous potential. Tremendous potential if we're able to combine the technology with the physiology. And so there's many people that have been imported here, Yuri Gerasimenko, Parag Gad, Joel Verdick from Caltech. You'll hear from this guy later on, and some of our Russian colleagues where we've collected a lot of data also. So, and there's other colleagues that have contributed. The work that Dan was talking about, we've been working together for about 5 years on that grant, and Lisa Moore has been one of the key players in that. And this is Susan Harkema, who we did the work with at University of Louisville with the first 4 subjects and her assistants. And we've gotten some assistance, but never enough. Thank you.
For one question, someone has a question? Yeah.
Great talk. I was wondering, in your talk and Dan's, you're talking about invasive and non-invasive techniques. And in the implanted ones, in those patients, were they not able to have restoration with neuromodulation that was external? Was that tried before the spinal cord implant?
The non-invasive started after.
Okay.
Yeah, the non-invasive technique started after, but not long after. And we're in the grant with Dan and I. We're testing both techniques. We're always asked which one's better, and we don't know. I think we need both of them, I think, to see how it works out.
Thank you very much, Reggie. So we're going to shift gears a little bit. Our next speaker is Dr. Wentai Liu, who Reggie just alluded to, who's been in the space of neurotech development and research for many years. Has been at UCLA for just over 10 years, something like that. And a lot of his early work actually translated into industry on visual prosthesis with retinal stimulation. So you'll hear his work in that space, I believe. And then I'm going to talk about cortical stimulation for restoring vision. And while we're getting that set up, I'll take a moment to comment. You know, Reggie made a great point about using, trying to reprogram the nervous system to use its smartness to rehabilitate patients. A lot of times what we do with neurotech solutions is we're trying to bypass injured parts of the nervous system. For example, brain-computer interfaces. That entails trying to decode and understand everything that happens between the brain and the effector muscle, which can be done and might need to be done in certain cases, but that's very challenging. I think the same thing goes for what Wentai and I are going to talk about, which is that if you stimulate at the retina, theoretically you're pretty early on in the visual pathways, and so much of what the brain does before it gets to visual cortex is a lot of processing that we take for granted that is important. So when we go back to the cortex to stimulate, there's a lot more challenges there because we've bypassed a lot of the pre-processing that's already occurred with that visual information. I think it's working on it. So keep that in mind. Sometimes, and then I'll keep talking because otherwise there'll be nothing going on. And something I've realized over the last 10 years in this field is sometimes the things that we think are the coolest are not actually what is the most useful. So it's really cool to do a brain-computer interface, and we learn a lot. I'm participating in a study, a trial with Richard Anderson, where we have a Utah array in a subject, and we're actually about to implant another subject, which is fantastic work. But the functioning utility may not be as great with something like that, right? So doing a lot of work from out of Cleveland as Case Western is on functional electrical stimulation, which is very cool to bypass the spinal cord, but that might not be the most effective way to do things. So we always have to counterbalance our enthusiasm and excitement for doing cool things with what is actually functionally meaningful to patients. And this should eventually open. I'm still ad-libbing. And we can have a discussion. Any other questions or comments?
Larger discussion at 11 in the whole morning session, but we can also take questions and comments now on the first set of talks.
Okay, all right.
Oh, you're right.
All right, so Wednesday was up to
Thank you. Okay, well, thank you already to introduce me, this guy. So I'm the guy. So I'm going to talk about the retinal prosthesis. And I think when Susanna and Neda approached me with, I'm interested to talk about visual prosthesis, and I said, yeah, okay. But I didn't underwrite to this. I have my personal story to say how important this training grant is. Because when I was an assistant professor, I was approached by two eye surgeons, and they told me that work with them and to develop this retinal implant. And I had no idea. Oh, I was purely electrical engineering. That's about 35 years ago. And then after that, then I struck into it, and I gradually changed my career. So I wish at time there's a training grant for me to pick up the knowledge in the medicine. So I trained myself to be able to speak two languages, engineering, and in addition to have very reasonable fluency in the medicine technology. So that's how important it is to have this training grant. Okay, so this is my talk outline. So when I talk about this retinal prosthesis, I'd like to give you a little bit history. So show that how do we start this project. And at the beginning, everybody said this is science fiction, but gradually that is evolving and become important. And even the White House notified this one. So I think that's how the interesting thing kicked out by ethical determination and so on. So here's the one. I think my lab has been working on various kind of implants. So ranging from the central nervous system to the spinal cord, because I'm part of Regis's research project. And I also have a very active one to target the autonomous nerve. So I think I want to give you a kind of overview. So of course my career started from here. I switched from electrical engineering to the biomedical engineering. And it turned out that it's good because my gray hair done run out into useless. So I do make some contribution. Then I've been working with Reggie to target the spinal cord. And recently I have a very active program to target the autonomous nerve system. So we do have an implant that can make the intestine or colon solve the POI problem. And those are the same technology I have developed in my lab. Over 30 years experience. So we can retarget and we can retune. And of course, there are also working on brain-brain communication. So I did in high-level stuff. And also I've been working with Dr. Fan in Northeastern University. So recently we published a paper in Science. And this one is set up in my lab. So we do make the transparent and stretchable. So this device we have implanted into the rat, the visual cortex V1 and V4. Then we can real-time monitoring what's going in the visual activity. And so we can do the two-photon imaging, and also we can do electro-physical recording. So let's just give you a... So my lab has been kind of... It's through some collaboration and also developing myself, so we are able to make all kind of components in the implant device, from the electro to the electronics, and even the wireless technology and internet technology. So we have make up a very special internet connectivity connection technology. We can connect the chip to either the electro and so on to preserve a very small footprint and so on. And we also have a very great expertise to develop the GUI interface. That means that the firmware approach. So I'm going to explain that a little later. So let me jump in back to this regaining the eyesight for the blind. And it's a kind of long story. So it ended up that this has become a commercial product. And I think this part actually was coming from my lab. So the engine for this device is coming from my lab. And that design was done in the year 2000. As you can see, it's 18 years technology. And what do we learn, and can we improve? So this is, I say this is plan A technology. Do we have plan B technology? And that's what I want to explain to you. Okay. So let me give a very brief story. So this project was started in research triangle. So we have three universities: Duke, University of North Carolina at Chapel Hill, and NC State University. So it happened that I was a professor at NC State University, a young assistant professor, and people come in and challenge me. I'm glad I took that. So I think we started this kind of project. So we have multiple people working on together. We do have a physician, and we do have a scientist. We have an industrialist, and myself as an engineer. So we got a lot of funding from NSF, NIH, and even Department of Energy. You ask me what the hell the Department of Energy are interested in this, because that's a sensor got involved. So to give you an idea, I think we target this special disease. One is called retinal pigmentosa, and the other one is called AMD, age-related macular degeneration. So when the disease progresses, actually, so anyway, when the disease progresses, this one becomes tunnel vision, and gradually the vision is gone. Well, this one is destroyed in the center. So this is the peripheral vision. It's gone first, and it's going to start from the central vision. Okay, so we started looking into you will engineer, we will design a device that such that we can kind of. Okay, so the disease, both disease destroy this piece of the layer in the retina. That's a photosensor, rod and cone, and both AMD and the RP disease as this phenomenon. So we're looking around, where can we put a device, in particular, where do we anchor the electrode? So we look into this location, or this location, or those locations. That's why we decided to go with epiretina. It's about the retina, and hopefully the electrical signal can reach those remaining neurons. And we also have a competitor that working on the subretinal approaches. So instead of doing here, they're doing here. And also we have the Australian and Japanese researchers. They start from here. So Germans take this approach. So I want to explain to you, why do we do that? We do have a competitor. So talking about visual prosthesis, as in, it can be said intraocular prosthesis or optical nerve, or we can jump start from the visual cortex. So we do have a competitor, epiretina. So starting from the Martin with Hopkins, Duke, and so on. Then second side joined later. Then we have Harvard MIT team. About a year later was established, then found in 1993. And Seoul University and Stanford University. Okay. Then I'm using color code. So the green one means it's established after 2000, but yellow code means before. Then there's another approach with subretina. So that's the green stuff we plug into the near the photosensor. Then also there's another approach. I think the Osaka University in 1997, Japan, they do this one, transretinal. So they put the electrode outside the eyeball. Then Australia government, I think they have special allocation, $50 million, and they start with for colloidal. So different location you can anchor your device such that you can stimulate the remaining neuron. And Virgin had to start this one instead of the retina. They start with the optical nerve. And here's the visual cortex. So now the second side, and working with the NEDA is working on this approach. But the Utah, Dick Norman and the Dover in New York University, they have been pursued this approach. So what's the reason we can do that? Because when this disease progresses, then actually the retina structure has been changed. Migraine, rewiring, and so on. Plasticity has been changed. So it all comes down to this phenomenon, this observation. So for the RP disease, the outer rod and cone is destroyed. You see that 46 verse 2. That's the majority has been gone. So there's no way to have a light sensation. But the remaining neuron, that's the that's bipolar cell, horizontal cell, American cell, are still, majority still preserved. Okay, and ganglion still preserved. Okay, So second one is for the AMD. You see that the outer layer is gone, but actually inner layer is still there. So that's the interesting hypothesis. Can we come out an electrical way to target this layer? And if yes, then we can build a device to make people to see again. So that's the early work. So I will show you that how exciting this one is. I hope I can run this one. This is to show the hypothesis. We put the electrode at the 9 o'clock position, and the physician move to 12 o'clock or 10 o'clock, and the patient can trace it. That's a very important step. The second one is that we have doing the temporal, so we can fire the 3 points in sequential way, and can patient distinguish those 3 points. And that's the device we use it. Okay, in that. So that's why on the engineering side we start looking at how do we design a device. So here's the idea: we design a device with the patient wear a sunglass, and we mount a camera there, and the visual field is coming from the camera, and we use electrical circuit to encode it into the Wi-Fi signal and transmit over to a receiver inside the eye. And then we receiver draw the pattern and the print on the retina. And this each dot is try to access the bipolar cell or ganglion cell because photosensor is gone. So that's the idea, and we started doing that design, and I think that's why we come up with this idea. And the first chip I have designed in my group is this one. And this one was the 0 generation one. But it turned out it's not that complicated, not sophisticated. So we started doing more design. And along the way, we convinced people to provide funding, and after about 10 years, we were able to persuade the NSF to give us the first grant. And I think we take spotlight, so I think the AP and there are about 50 kind of news media come out to have a press news, and I took this opportunity. So Dr. Humayun and myself, we are talking to this first patient, and that's the idea. Then even the president recognized this one. So I think in the 2000 years of the State of Union address, special mention about this project, and also I didn't mention about project Reggie is doing. So I think it happened, I'm lucky, I'm involved in these 2 projects, and I think that's what predict in the year 2000. So I want to show you. So I think, looking back, I realized that the federal government, the federal agency play a very important role, because that this is too brave project. Usually that we cannot get funding from the other outside of the federal agency. So at the time we are lucky, Dr. Gia Devi is a visionary bioengineering. So especially like this project, even people, the reviewer said this is a daunting project, but he said, okay, I'm going to fund it. So I think we got the funding from that. And then after that, he helped us to establish an engineering research center. It's a 10-year program, and that's why I think it's kicking up after President Clinton's speech. Then we also have Department of Energy coming in. It's an 8-year program and support. So Dr. Dinko, and of course there's the NIH BRP project kicking in. So I think those are important. So early work, so engineering work, so we're starting from the first generation, second generation, third generation, and fourth generation. So this is one mandated by NSA ERC. So we got to design a 32 by 32 pixels. So this one is 4 by 4, then 8 by 8, and 16 by 16, and 32 by 32. I think I don't have time, so I can run this. Okay. So each generation has its own characteristics. So this is the first generation electron, this is the second generation electron, then third generation electron, and fourth generation electron. So first generation, the chip was developed in my lab, and this is done in 2000. So this is the foundation of the second size, August 2. Okay, and this is DOE's project. This is our NSF. So we do have developed that, and I think I took this opportunity. We do have some meeting together, so I was able to take some picture with the real patient in New York, Chicago, and Palo Alto. Okay, so I think very quickly to show you that the first generation, this is the first generation with 4x4 pixel. You see, can I run a movie? And this is the second generation. So the response time is much shorter because we have 8x8, and this is the R+2. Okay. Then I think we have done this in the real environment. So this is experience in the Paris. Sorry. So this is the second size device. Okay. And I also take this opportunity that introduce this device to the Taiwan, my homeland country, and I personally participate in the experience of this device. So we do see that kind of excitement on this. So now the second size device has about more than 300 set commercially in print. And so I'm showing the industrial player. So now we have second size device, and we also have the German Alpha MS. And by the way, this has been approved by FDA, only the early feasibility study. So it's still a long way to get FDA approval. And then we have the French company Pixin. So they are all very similar design, very small difference. And this one is Australia. So we have 4 major competitors compete with second size. But we're looking back, what can we do better? Because that's plan A. So I think all those 4 company devices, in my opinion, it also have a major deficiency. And what are the deficiencies? So that's why I want to look back. There's a new physiology inside, and even a new technology come out. So I think if we want to do a better job, it's very similar to the cochlear implant. Cochlear implant was initiated in 1964, but now people still working on the cochlear implant to improve the efficacy and efficiency. So I believe that we are following the same pathway for the cochlear implant. The retinal prosthesis need to be improved, but the retinal prosthesis improvement really heavily depend on technology. So I want to show you what the potential problem with the current device. Number one is there's a spurious light spot. I think the patient complained that after the implant and they see a lot of dots. It's like in the nighttime you look at the star, and those are not really the image you want to see. So I think how do we avoid that? And I think the new physiology study shows instead of stimulating with 0.5 milliseconds, we can stimulate with say 20 milliseconds to 25 milliseconds. So stimulate with a longer time can resolve that issue. But the current device, all those whole major companies, they don't have this way. They're using counter, they only count to about millisecond. So how to support 20 milliseconds, 21 milliseconds, even 100 milliseconds. So that's one of the major obstacles. Second one is that you continue to stimulate and you're going to cause the neuron to be fat and to fatigue and fat. So I think that's the reason that we need to initiate some randomized stimulation. So instead of regular reflection rate stimulation, the device must be able to do randomized or even emulate the biosignal stimulation. So in our body, we have some biomimetic signal reason, and we should stimulate based on that, and it can increase the effectiveness. But the current device cannot do that. And also crosstalk is a major issue because we want to increase the number of the electrodes. And when we increase the number of electrodes, the distance will be reduced, and that crosstalk, electrical field will cross over. So we need to work on other techniques. So the one of the technique is called virtual electrode idea, or using spatial filter idea. So all those kind of technique currently cannot be implemented in the current device. And that's why my lab is working on that, so to be able to do that. And of course, the most wishful list is color vision. Can we provide patients with colorful vision? And I think this also has been worked out. So I'm using my colleague, Jim Wayne, my good friend, for almost 30 years. Recently, he published a paper in Nature. they're showing that you stimulate with a short pulse, with long pulse. The long pulse provide a better focus. And that's why I think the field is moving toward that. If we can have a device that can provide a longer pulse, then we can provide better focus. And this is another issue. The one, if we're changing the electrode size to very small, then apply with a longer pulse, and then I think the focus ability is increased. And the third one is that if we do that, then see this is the case, lift. Okay, so using the current device, you will see a lot of spot like this, and that's why the patient complain. Got to do a lot of training. Psychophysics tell them, okay, to remove those, but I think that's a very difficult task. So that's the problem. And okay, color vision. So I think we know how to do color vision, but current device cannot support this. So it requires a lot of sophisticated encoding, and that's why we need a new device to do the encoding. Okay, then randomize. So we got to bring the randomize. So this is the curve show that this Australian group show that if we can do randomize, then the spike rate is higher, and so it's less fat, and fatigue is less severe. Okay, then of course Maile has developed the virtual electrode idea. Okay, pretty quick. So what's next? So that's why I'm at UCLA. I'm looking on that. Can I develop a Plan B device and be able to accommodate all those wishful list? And I believe I've been successful to do that. So I think I'm working on an upgradable architecture. So I think I have a platform technology implanted, but it has been carefully designed. So it can changing the stimulation paradigms from externally. So we call this an upgradable architecture. And I do have a sample here. Okay. So this is a device that we have implemented, and you can externally reprogram it to support all those features: longer pulse, randomized stimulation, colorful vision, and so on. So offer a very short attribute. So I think the success of this kind of interdisciplinary project is that we need to have a very good team players, and we got to have a visionary officials at the federal agency because it's important. And we got to demonstrate creativity, determination, and perseverance. So I think for the training grant, see the interdisciplinary research is very rewarding, but it's long and frustration. So I've been honestly, for 30 years, I feel frustrated, but be patient. And second thing is that the university growth really, it's just too thin to carry such a big burden or task. So with that, I'm ending. Thank you.
Any questions?
One question while he sets up the next step.
Okay. All right, so I'm going to speak on the other end, and which is at the cortical level of stimulation to restore vision. This is a project that has been underway for about 6 years, and interestingly enough, the way I got connected with Second Sight was not through Wentai, even though he was here when I first started working with them. Was because one of my fellow graduate students, when I did my neuroscience PhD here, became the director of basically research at Second Sight, and when they were looking for a collaborator, found me at UCLA, and that's how we started this connection. So it was a grad school connection that got me to this point and interested in this topic. The technology that we use, or that Second Sight is using for this research, is adapted from the early generation work that Wentai did to develop the retinal prosthesis. So in designing the cortical prosthetic, the goal was to use the technology they already had, adapt it for cortical stimulation. And that speaks to what Wentai mentioned, which is that you don't need to create a completely new device for every application. There's a lot of crossover. There's a lot of similar underpinnings to stimulation devices, whether you're stimulating at the retina, spinal cord, or the brain. Okay, so just to put things in perspective in terms of blindness, there are 39 people 39 million people worldwide that are blind. And as you heard, the retinal prosthetic that's currently FDA approved only serves patients with retinitis pigmentosa. They're looking at age-related macular degeneration, but that's another 2 million people, and other untreatable causes of blindness is about 6 million. So this is a very small fragment of the population. Hopefully, this retinal prosthetic that is approved will be able to be used for other patients with blindness. But the goal here was to turn to perhaps a different site of stimulation, right? So the light will will go through the cornea, hit the retina, go through the optic nerves, through the tracts, back to the thalamus, and from the thalamus back to the cortex, and then it's processed from there. And so there's multiple areas where you could potentially intervene in the visual system in order to restore vision. Our goal was to develop a cortical prosthetic, which dovetails nicely with my clinical practices, which is brain stimulation for movement disorders and other applications. But in developing cortical prosthetic, I'm going to give a little bit of a clinical perspective here. You know, it's great that you can design a device that may work, but there's also other concerns, right? So there's safety concerns. How do you design an implant that is easy, compatible, easily adoptable, right? So you don't want to create an extremely difficult device to implant, because then if you have it, if it can't be clinically adopted, then it's useless. There's a very significant concern with cortical stimulation with seizures. If we are simulating with 60 contacts on the cortical surface, that is a great way to induce seizures, and so we had concerns about whether or not we can do this on a continuous basis and not have patients have seizures. Practical concerns about, again, like I said, is surgically practical and adaptable. And then clinical utility, perceptions, whether what you perceive is useful, whether you're going to see shadows, edges. You know, I think when I started this project early on, I thought, wouldn't it be great if we could restore vision the way that we all know vision to be? But again, getting back to Dr. Dobkin's point, we don't necessarily need to do that. Right? So to restore function or make someone function or help someone be functionally rehabilitated, they may need a very small fraction of what we need in order to be able to reintegrate in a functional way into society to fill those gaps that they might have lost. And so the work I'm going to describe for you really focuses on this aspect, the clinical utility, what people perceive with stimulation, how stimulation parameters affect the perception. And as noted on my initial slide, this is a lot of the work done here. All the work was done in collaboration with Second Sight, but also Sarush Nikatharad, who's a graduate student with me, has done much of this work. This is not completely new work. There was early work done with, this is an array that's implanted over the occipital lobe. So the medial occipital lobe is where the visual cortices are. And it has been mapped out so that if you stimulate a certain area, they were able to demonstrate that people perceive things in a certain area of their visual field. And this was done relatively reliably, but with an externalized system with arrays with connectors sticking out of the skin and not over a chronic period. And these research efforts pretty much, unfortunately, ended with without much of a clinical product at the end. And so we really tried to pick up on this work. There's a lot of research being done, and to note a opportunity in neurosurgical patients, in patients who are undergoing monitoring for epilepsy surgery. So this is out of Stanford University. A lot of work is also done by Dan Yosher and Michael Beauchamp at Baylor in terms of understanding how sighted individuals will see or perceive stimulation of the visual cortex. Now, there's a big difference between testing in sighted individuals versus blind individuals, and it's not necessarily true that it will translate what we see in sighted individuals to blind subjects, but still this work is really important. Briefly, they only had, I think there's 5 electrodes here that they assessed, and they looked at the size of the perceived phosphene, and you can see as they increase the current, it seems like the size has nothing to do with the current, which is not totally true, because the amount of cortex dedicated to each area of the visual field is different. And if you look at this, how large a cortical area is needed to evoke a phosphene of that size, if you correct for the phosphene within the visual field, there's actually a pretty tight correlation in terms of how much cortex is activated by the amount of charge that's deposited, and so you can get a pretty good and accurate estimate of what someone will perceive based on what we know about the mapping of the visual cortex, or how the visual fields are mapped onto the visual cortex. Again, that's in a sighted subject. So when we started this work, we were really, although there was some work that had been done in blind subjects, and although there had been work done in both blind subjects and sighted subjects, we decided that we needed to start anew and really get a very good handle on the stimulation parameters that affect visual perception. And so we did a N of 1 study, which means we had a single volunteer. It was a 30-year-old young lady with a 8-year history of bare light perception. She had a little spot of light that she can see. And blindness due to this condition, which is Vogt-Koyanagi-Harada syndrome, which is an autoimmune and inflammatory syndrome. And we took an off-the-shelf neurostimulator, which is the NeuroPace system, which is a responsive neurostimulator. We actually weren't using the responsive factors in this, but a device that we can implant in the skull and then implant 2 arrays over the visual cortex. And this would be the first chronically implanted, fully implanted system over the visual cortex for testing visual perceptions. So this is the patient's brain. These are 2 arrays that we implanted over the visual cortex. That's where our arrays ended up. And this was our test bed for over a year of testing this individual, usually about once a week, testing different parameters of stimulation, different frequencies of stimulation, different pulse widths, many different stimulation parameters. And these are the perceptions that she has. So she would fixate, we'd have her point with her finger to the center of a screen and imagine that she's fixating on that point. And then we would turn on an electrode and we'd have her draw out what she would see. And you could see what she would see was a large phosphene here. When it's closer in, as you'd expect, when it's closer to your center of vision, the phosphines are smaller, which is what we would expect. So the further away it is, the larger it is. Relatively reproducible. So you can see there's multiple shadows there. Every time we turned it on, it was in the relatively same spot. Now, something we quickly became aware of, things that you don't think of ahead of time when you start thinking about clinical translation, eye movement actually makes a huge difference. So when you see something flashing in this part of your vision, you tend to look towards it. It turns out when you look towards it, it moves the phosphene further and further away because everything is centered based on your center of gaze. So we really need to coach these patients or these subjects to fixate their gaze and not move their gaze. The same issue arises with a retinal prosthetic as well. So within sessions, she has very consistent perceptions. Here's the same contact over and over again. Here's another contact. There's some variability here, presumably due to eye movements. This is the most variable one that we see here. But we also see reproducibility over multiple sessions. So you can see clustering of these perceptions over time. So this reproducibility is extremely important because if it's not reproducible, then you're not going to be able to reliably advance a cortical prosthetic for restoring vision. The other thing that we did a lot of experiments with different stimulation parameters, different, as I said, pulse widths, frequencies, and other stimulation parameters. And what we saw were 2 phenomena that has been characterized at the neural level, so the individual neuron level, that came out with macro stimulation. So you wouldn't necessarily think that stimulating, with a 4 millimeter contact would have the same stimulation properties as neural stimulation, but it does. And what we saw was a Rio-based current, so there's a minimum amplitude with the infinite pulse width frequency that's present. So it doesn't matter how large your pulse width is, there is a minimum amplitude that you need to be able to deliver in order to get the perception of a phosphene. And that's with frequency modulation as well as pulse width modulation. And look at it a different way. So there's a minimum charge that is required to elicit a phosphene. So it doesn't matter how small your pulse width is, there's a minimum amount of charge that you're going to need. And anything above that basically is you're dumping extra charge into the patient's brain that doesn't necessarily translate into a perception. Or it doesn't change the perception, is not needed for the perception. But we do get modulation of the perception with increasing the frequency or pulse width. So, and it affects it differently, interestingly. So even though the total charge per trial, for example, right here, might be 10 microcoulombs, by getting to 10 microcoulombs by modulating frequency, it has a different effect than modulating the amplitude of the stimulation. So these are things that we hadn't known before, but the subject, by being able to test her comprehensively, really helped us gain insights. As you can see also, there's a certain maximum to the perception also with frequency modulation. You can keep simulating at higher and higher frequencies, but that doesn't change the perception. But increasing the amplitude does seem to continue to increase the perception of stimulation. So what we got from this is a couple things. The V1 stimulation, so cortical stimulation, is feasible for vision restoration. And I'm going to talk about this in a moment, but you'll notice what we were showing were not fine features, right? You can't imagine putting those all together to restore vision the way that we know vision. But the good thing is these phosphines that we do see are very predictable over time, and they're based on basic science insights into the physiology of neuronal excitation, and these phosphines are reproducible and consistent over time. So we really are doing more studies, and we've actually moved on from this single subject. And I'll tell you a little bit about the clinical trial that we're in, the early feasibility trial. But we need to understand phosphine modulations. How do we achieve different shapes? How do we connect different areas of stimulation to perceive shapes? How do we change the size? Color is a big deal. Color is a very emotional component. We underrate what it is. But with this subject, the first day we turned her stimulation on, she saw color, and that was the first time she became emotional about the experience, which brings into the question the whole ethical, the ethics of what we do in terms of restoring function and the limitations and considerations that we need to consider. Temporal factors, so what happens with sequential or simultaneous? Can we assume that stimulating two different contacts automatically means that you're going to sum those two, or you're going to get something in between those two contacts in terms of the phosphine perception? What happens with sequential stimulation, or when you scan across multiple electrodes? Does that result in a perception of a moving target or a static object? So how do we create shapes with temporal factors? Frame of reference, head and eye position is extremely important. And then we need to do studies with purpose-specific devices. So this is the only cartoon rendition that's been released by the company. Company's a public company, so we can only disclose what they've disclosed. But it's an adaptation of what Wentai showed for the retinal prosthetic. This array here is actually triangular, and it's meant to cover the cortical surface here in the visual cortex. And it still uses the glasses with a video camera and a video processing unit that then wirelessly communicates with this stimulation device through an RF coil. The tests here are really to understand the feasibility, utility, and consistency of the response to chronic epicortical stimulation. I should say the surgical technique to do this is actually, in neurosurgical speak, relatively easy. Still brain surgery, but it's not that hard a surgery. It's within the normal repertoire of surgeries that we would do. It's not that different. I will say it took us, I started this by saying this has been 6 years in the making. We didn't get to implant our first subject until January or February of this year. It takes a long time to get to an early feasibility study. We spent a lot of time with cadavers. We did a lot of large animal studies and other feasibility studies and multiple iterations of this prosthetic before we actually got it into humans, with very close collaboration with the FDA. And we've implanted 5 subjects across 2 sites to investigate this feasibility. We've we've had people use the glasses. They can consistently see spots of light. It's very chronic, very consistent stimulation with chronic use. People have been using this with up to 2 hours of continuous stimulation. So the visual cortex is being continuously stimulated for 2 hours at a time. And we've now started letting people go home with the device so you can see what it's like at home and for their use. That's what the device looks like. That's what the implant looks like. It's a little bloodier than I like it to be, but it's a small little opening in the skull. That's the device implanted on the skull, and then it goes into the inner hemisphere fissure. We've made this incision a little bit smaller in subsequent surgeries, but it's still a fairly large incision to get that in place. So I want to think, yeah. So I want to, how much time do I have? Okay. I talked about these issues. So the interesting things that come up. I told you that we know a lot about the spatial map of the visual cortex. We've been using, a lot of questions about whether we use what we know about the anatomy or whether we map things out with individual subjects and to find out where they're seeing phosphenes. How reliable are subjects versus how reliable is the neuroscience underlying visual cortex anatomy? This is an extremely important point. Rehabilitation is, again, this is like that transition I had from we're going to restore normal vision to we're going to restore spots of light. I didn't think about what rehabilitation meant in the sense of developing a prosthetic. It is so important because I don't think in our generation or in our lifetime that we're going to restore vision the way that we know vision to be. It does not mean that it's not going to be useful though, right? So we have to get out of the mindset that we need to make people normal again. We need to make them functional again. And so the rehabilitation is important. I'm excited about them going home with the device because I want them to go home and tell me what is it that they want to do, what is it that they can do, and how do we bring those closer together? Where are the obstacles? And so we have visual rehab specialists going to these people's homes and working with them to figure out how do we overcome the functional obstacles that they need, even if it doesn't mean normal vision. I just saw a fascinating talk by, I just blanked on his name, David Eagleman from Stanford on the idea of sensory substitution. Just because we see and we know the world as we know it with the various types of sensory stimuli that our body has been adapted to as humans, it doesn't mean that we have to be limited in scope in terms of being limited to restoring that kind of function. We can try and use different types of sensory input to restore function. And last, neuroethical issues. There's a lot of issues that come up that you can't even begin to imagine when you first start this type of project. You need to be very cognizant of them, but it's exciting. One last thing, the funding for all this. It's an amazing time to be in this space because there's a lot of money being thrown at it, especially by the NIH and DARPA and everyone, NSF, because of the BRAIN initiative. So they're very keen on developing new devices, new applications, and understanding the neuroscience underlying the types of neurotechnology that we want to develop. I'll conclude there. Thank you. Any questions?
Not? Oh.
Yes.
Is the vision that you're eliciting with phosphenes, is it shades of gray? I mean, is it more aimed to the edges or?
So they are consistently seeing spots of light, but they can also see movement of light, right? So they can see moving grading. So when they put on their glasses, they can navigate around a room. There's this bright light in the back of them. They'd be able to easily navigate towards the bright light in the room. They can, in some cases, in many cases, they can identify the direction of movement and a grating that's moving in front of them. So they can see, it's meaningful sights, just not the sight that we're used to. But now it's a matter of teaching them to interpret what they're seeing because it's not the same vision that they once had. All right, we will take a short break and then we'll reconvene. Thank you very much.